In this lecture video, we are going to learn about the first part of the cardiovascular chapter, blood. Okay, so let me get the PowerPoint slide and then we can begin. So your cardiovascular system, we can learn in like three parts. So blood, which is the fluid that roaming around in your body. in the vessels called blood vessels, and we are going to learn about blood vessels, and then and also the pump, which is known as heart, which is pumping this fluid through the blood vessels that roaming around in your body. So we have distinctively three areas of this cardiovascular system. So one of the largest systems that you're going to study in this semester. Okay, so this chapter deals only with the fluid that is going through the blood vessels and doing their work. So blood is a connective tissue. Now in ENP1, you learned that we have four types of tissues: epithelial tissue, connective tissue, muscular tissue, and nervous tissue. So tissue means group of cells, but connective tissue, as you learned before, that it is not only a group of cells, but they do also have some non-cellular material, commonly known as matrix. So in this case, blood also follow the same strategy. They have cells, which we are calling them formed elements. I'm going to mention them detail a little bit later. And they do also have matrix that is known as blood plasma. So blood is considered the fluid connective tissue based on their state. Okay. They have tremendous amount of function in the body. The first function is blood is acting like a limousine service, like a taxi. Okay, so they are carrying stuff. So they carry gases like oxygen, carbon dioxide back and forth from the cell to lung, lung to the cell. They carry nutrients, they carry hormones. They carry metabolic waste, like all the waste product that you produce. They carry multiple enzymes. They carry a lot of important factors that are involved in different type of reactions, such as inflammation, such as blood clotting. So they carry many things. Okay? They also regulate certain things. So they regulate the blood pH. And they regulate the electrolyte balance in the body, like in ionic composition, like how much sodium, how much potassium, how much calcium you will have, how much bicarb, how much chloride ion you will have. All these things they regulate. Okay, so they regulate in conjunction with the kidney, like what to keep, what to let go. So that we are going to learn a little bit more in the urinary system as well. Then they restrict the fluid loss. What does that mean? Blood clotting. We all know that when you get a cut in the blood vessels, the blood clot after some time, so that does not allow the liquid to go out of your body. Because remember, it's a closed circulation. We have a closed circulation in the body, so that they don't allow liquid to go outside. Okay, we have a fixed amount of blood. Yeah, we're making it every day, we're replenishing it, but we don't want to lose it. So blood clotting is the one. Defending the body. So blood contains, let's say, for example, white blood cells. They defend your body. So they defend your body against toxins, against pathogens. Okay, blood has blood can carry inflammation. You're going to learn an immune system. That's also one way to defend the body. Blood contains a lot of antibodies which work against antigen, defend your body. So defense, more detailed mechanism we are going to cover in immune system class. And finally, by distributing the heat that mostly muscle, let's say, produce, or a different other chemical reaction that produce heat, by distributing them throughout your body, blood is controlling your body temperature. So they regulate the body temperature by redistributing the heat, or sometimes they absorb the heat. So the blood has a lot of roles. So how we are going to approach this chapter? We are going to learn that what is in the blood. So like composition of blood. So once we know the composition of blood, and then we are going to approach individually. Like these are the cells in the blood, these are the proteins in the blood. So what are these things does? What are these things that? And then we are going to talk about like how we produce blood cells in the body. And we're going to talk about blood clotting a little bit separately, and we're going to talk about blood typing. Okay. So that's basically the entire chapter. So blood, as I said, it's a connective tissue. It has two parts. So the liquid part, the fluid part is, or matrix, in this case, is called plasma. So blood plasma is like a straw color, like a light yellowish color, or like towards the clearish side of a liquid. And formed elements, basically these are cell and cell fragments. That's why we call formed elements, because some of them are not technically cell. They have like a fragment of a cell, like platelets. So they have just a membrane bound structure. So we call them formed elements. They are called red blood cell or erythrocytes. You should know both the names. Red blood cell, RBC, or erythrocyte. Erythro means red. Cyte means cell. Erythrocyte, red cell. White blood cell, leukocyte, same thing. Leuko means white. And platelets, thrombocytes. So these are the cells in the body. So again, how we are going to approach? We are going to learn about plasma first, meaning what are the components in the plasma and what they do. And then we are going to talk about the formed elements like RBC in detail, WBC and platelets in detail. Okay. This is a blood slide looks like under the microscope. Okay, so when you're looking at a blood slide, and I'm sure you have looked, if maybe in your lab class you have looked at some of these blood slides or some pictures, they appear like this. Okay, so these are millions of those red blood cells. Those are tiny donut-shaped bubble without nucleus. And the little bit larger cells with the nucleus, those are WBC. In our body, we have different type of WBC. We're going to talk about that later on. And this fragment, cells fragment, the small one, those are called platelets. So if you think about blood in two parts, one part is blood plasma, the other part is formed elements. The first question is how much percentage of that whole blood is blood plasma? It's obviously very. Same thing with formed elements is obviously very. So plasma is approximately like 46 to 63%. Formed elements is like 37 to 54%. Instead of memorizing those, the range, or what we can do, we can go to the middle. Like we have approximately 55% plasma, approximately 45% of the blood cells or formed elements. So your plasma is slightly more than 50% in the blood. Now, in the plasma, we have 92% water. So plasma constitute mostly of water, and they have approximately 7% of protein, different types of protein, and 1% other molecules. So when you say others, other could be electrolytes, let's say sodium, potassium, calcium, phosphate, sulfate, bicarbonate, chloride, magnesium, any kind of electrolyte or ions that you can think in your body. They can have organic nutrients. They can have organic waste. So they can have urea, uric acid. Those are waste. They can have nutrients like like a lot of like glucose, for example. The carbohydrate that you eat, glucose, lipids, different type of lipids. Okay, amino acids, cholesterol, all these things, and even hormones, and all these things could be in that other nutrients. Okay. Obviously, it's not possible to memorize all the word like names, but just to get an idea what other solute means is good. Now, coming back to protein, we are going to focus on three major protein that in the plasma. Although technically, most books divide them into four different type of protein. So albumin, which is the most abundant, 60% of the protein concentration is albumin. Then globulin, which is approximately 35%, and then fibrinogen is approximately 4 to 5%. Now, the remaining 1%, we categorize them as like a regulatory proteins. What that does mean, meaning technically some enzyme, the enzymes are protein, most enzymes are protein, technically most proenzymes are protein, technically some hormones are protein, and they are in the blood plasma. So we can consider that as a group of protein, but they're less than 1%. So we are going to focus now like albumin, globulin, fibrinogen. Those are the three major variety of protein proteins in the plasma and what their function. So the most abundant is the albumin. So what their job is, their main job, they have like two functions mainly. One is they can maintain the viscosity and osmolarity of the blood. So they are the main contributor to the viscosity and osmolarity. And they also carry stuff, meaning they are responsible for transporting a lot of molecules. Okay, so their albumin has two major functions. One is to help transporting other molecules. Two, maintain the viscosity and osmolarity. So what is viscosity and osmolarity? It's basically, in simple word, how thick should be your blood. That maintained by albumins. Okay, so albumins, like if your blood is too thin, like water, they will flow too quickly. So they will, remember, when blood flows through the capillary, like a narrow blood vessels, those from molecules from blood that goes into the cells, into the tissues, so that they can receive the nutrients and they get back the waste product. That is like osmolarity. So based on the osmolarity, those things, molecules can travel back and forth outside the blood vessel, coming into the blood vessels. And viscosity is like how thick your blood is. So if it is, as I said, is too thin as water, it's going to move too fast. Or if it is too thick as, let's say, honey or like mineral oil, and it will move too slow. So we need to have a little bit of viscosity, like it cannot be as thin as water, but it cannot be as thick as honey. So that maintenance is by albumins. Okay. And obviously albumin transport like different lipids, different hormones. Albumin is a great transporter. Globulins, the second largest variety. Globulins, different type. They are according to their structure. They're like alpha globulin, beta globulin, and gamma globulin. What alpha beta does is same as albumin, meaning they involve in transportation. They can transport lipid, they can transport metal ions, etc. Okay, what gamma globulin are, they are the basically antibodies. So they are the antibodies that actually work against the antigen. So they help defend your body. Okay, detailed structure about gamma globulins, how they produce, how they work, we're going to learn in the immune system. Right here, we just need to know that gamma globulins, one of the globulins that provide defense for your body. And alpha beta, they transport. Finally, the fibrinogen, the protein that is responsible for blood clotting. So we are going to learn blood clot in detail later on in this chapter. So blood clotting, one of the major player is fibrinogen. It is a very complex process. We are going to learn that at the end. Now, once it is clot, let's say that you collect the blood and it clots. If you remove that clot, the remaining fluid, we call that fluid serum. So serum is a remaining fluid when you remove, let's say, fibrinogen. So in the absence of fibrinogen, that's a protein, the one that is responsible for clot, the remainder of the plasma, we call it serum. It's very important for clinical use because it contains a lot of other molecules and other stuff that is useful, for example, antibodies, which is useful for a lot of clinical purposes. Now, if you look at the formed elements, the cells, okay, 45% of the blood is cells. In that cells, like red blood cell is like 99.9%. So they are the majority. And then like 0.1% is like platelets and white blood cells. So they're in thousands, while red blood cell is like millions. So like platelets average is like 250,000, like 130 to 400,000 approximately. So 250 average, and white blood cell in like 5,000, 3,000 to 5,000. Red blood cells in millions, like 4 to 6 million per microliter of blood. So they're a lot. Okay. That is why it appears, the blood appears red, because they are the majority. So before we go into the individual like cells, like red blood cell, white blood cell, platelets, what they do, let's talk about a little bit of like how blood is produced. So every single day, the blood is being produced, like 400 billion of platelets, 200 billion of RBC, 10 billion of WBC every day we're producing, because the rest is being recycled. So we are recycling the other and producing them. The production of this blood, particularly the formed elements, like all the blood cells, is called hemopoiesis. Poiesis means production, formation. Hem, blood. Hemopoiesis, blood production. Now, hemopoietic tissue, meaning the cells, the cluster of cells that are responsible for production of the blood, they are actually, during the time of fetal development, they're in the yolk sac. That, typical yellow color sac that present, that's right there. From those yolk sac, the first group of stem cell, they go and, they settle down in, let's say, bone marrow, in the liver, the spleen, and the thymus. These are the organs. So they settle down in the liver, spleen, thymus, and bone marrow. But, liver stops, like at the time of birth, the liver cells is not responsible for production of blood cells. And spleen and thymus, they get more involved into the leukocyte and lymphocyte mainly production. And we are going to talk about that later. But then bone marrow becomes the main place where the red blood cells and most of the other white blood cells are being produced every day. Quantity of the erythrocytes and hemoglobin. So basically, this is to understand that, like in general, the main function of blood is to carry oxygen. The main function. All functions are important, but the main thing comes into your mind when you're thinking about blood, that they carry oxygen. Now, in order for blood to carry oxygen in proper way, you measure three things to understand. One is called hematocrit, two is hemoglobin concentration, and three is RBC count. All three are directly related to how the oxygen will be carried. Okay. So hematocrit, which is called packed cell volume, or some book will call volume of packed red cell. So basically, this is a measurement percentage of whole blood, percentage of RBC in the whole blood. So how much RBC you have percentage-wise in the whole blood. So what in the lab, what we do, you can take a test tube, or nowadays we take like a capillary tube, so you don't need too much blood. So if you take a test tube, and if you like a whole blood, and you spin them, called centrifugation. So if you centrifuge them, if you spin them, we know the heavier particle will settle down in the tube. So if you look at the tube, the bottom part will have all the cells. The top part will have that clear plasma. That bottom part, that percentage of the entire thing is called hematocrit. It's basically almost entirely composed of red blood cell, maybe on the top a little coat of white blood cells and platelets. So like 99.9% is the red blood cell. That's called hematocrit. Okay, so we can ignore that top buffy coat of WBC on that packed volume. So in case of men, it's like 42 to 52%. In case of women, a little bit less, like 37 to 48%. Okay. So that can give a direct information to the clinicians or healthcare providers that whether your blood can carry enough oxygen to be remain healthy. Same thing with hemoglobin concentration. Whole blood hemoglobin concentration, men has like approximately 13 to 18, women a little bit less, 12 to 16. Same thing with RBC count, all between 4 to 6 million with a little bit vary. So there are like three physiological reasons why the women has a little bit less than men. Because reason number one is, the androgen, like the hormone testosterone, the androgen, they influence the blood cell production more. So that could be one reason. And reason number two, it varies inversely with body fat. And reason number three, this is in general, like as a whole. Reason number three, if the woman is in their reproductive age, they lose a lot of blood during menstrual cycle. So because of loss and because of the production, generally, overall, obviously individual variations are there, men tend to have a little bit higher hematocrit value, higher hemoglobin concentration, and higher RBC count. And all these three, hematocrit, hemoglobin concentration, and RBC count, directly influence the amount of oxygen the blood can carry. Now, the structure of the RBC, like how they look like. So red blood cell, they appear as a biconcave disc. So basically biconcave means in a little bit narrow in the middle. So the biconcave on both sides, they come like a little narrower shape. So their middle, they become much more narrower than in the edge. So if you imagine like a donut without that hole in the middle, that's your red blood cell. It's like a disc, and this structure helps them to move like a squeeze through. They're a little bit dynamic structure, so they can squeeze through those tiny capillary as well. But what interesting about red blood cell is not only that. The interesting thing is about that they don't have a nucleus. So they do not have the ejecta nucleus when they're undergo formation. We are going to talk about that soon. So they don't have nucleus. So obviously no nucleus means no DNA, and no DNA means no RNA, and thereby no protein synthesis. So red blood cell does not divide, so they don't undergo mitosis. They do not do protein synthesis, and they also do not have mitochondria. And mitochondria, in the cell chapter, probably in NP1, you learned that mitochondria is directly responsible for production of maximum number of ATP in the body. And that does not help. So they don't have mitochondria. So what red blood cell does, they get ATP only through anaerobic fermentation or anaerobic respiration, meaning without mitochondria, you cannot do, if you do remember, like Krebs cycle or electron transport chain. Without mitochondria, that is not possible. So you can only go from glucose to pyruvic acid to lactic acid. That's happened in red blood cell. Approximate lifespan is like 100 to 120 days. So it's like leasing a vehicle. So you lease a vehicle for 3 years and you return and get a new one. Same thing. They take blood cells, the body form them. They're like 3.5 to 4 months. They're actively performing, and then they have a wear and tear. Body takes them away and recycle them. And we're going to soon learn how and what they do with the recycling. So what red blood cell is mainly responsible for? The gas transport. So they take oxygen from lung to the cell. They take carbon dioxide from cell to the lung so that we can release them out. Okay. So detail about gas transport, like exactly where oxygen, how it binds to the hemoglobin molecules, and how the oxygen is carried, and same thing with carbon dioxide. This whole thing we are going to study more detail in respiratory system. So you can ignore this particular part right now. Like how exactly they're being carried or called gas transport. We're going to talk about these carbonic anhydrates in respiratory system. So hemoglobin, that is the protein that we're talking about. So your red blood cell is kind of like a packet full of hemoglobin. So it's technically, a red blood cell is a packet that is completely composed of, let's say, hemoglobin, because they don't have nucleus, they don't have mitochondria. So they are like a packet of hemoglobin molecules. They have like 250 to 280 million hemoglobin can be packed in a red blood cell. So now, what is hemoglobin molecule? Hemoglobin molecule consists of 4 polypeptide chains. We know the protein molecules are nothing but polypeptide chain. They have 4 of them. 2 chain we call alpha, 2 chain we call beta. That's a typical normal adult hemoglobin. Okay, there is a slight variation in the fetal hemoglobin. So fetal hemoglobin, instead of 2 beta chains, they have 2 gamma chains. So beta and gamma, they're almost same, only gamma chains, the hemoglobin with gamma chains, which are called fetal hemoglobin, can have a little bit more attraction towards the oxygen. So that's why they can literally, like if you think about the word snatching, they snatch the oxygen from the mother's, so the fetus can take the oxygen away from mother's blood to them. So each adult hemoglobin, they have like 4 polypeptide chain, 2 alpha and 2 beta. Now, this is important, at the end of each chain. So in this picture, like there are two alpha chain. This blue color one is alpha, and the golden color one, that's beta. So at the end of each chain, this red star, that's a molecule, that's a moiety. That's not a protein moiety. That's a non-protein moiety, which looks like this huge organic molecule. And at the center, they have an iron atom. The iron atom, ferrous, the iron atom right at the center. So oxygen literally binds to this iron atom. So each iron atom can bind one oxygen molecule. So technically speaking, one hemoglobin molecule then bind with 4 oxygen molecules. And if we have 280 million hemoglobin in a red blood cell, so individually each red blood cell can carry 1 billion molecule of oxygen. So it's a great carrier of oxygen that helps our body to maintain the demands. Okay? So bottom line is, what makes hemoglobin? A hemoglobin molecule consists of 4 protein chain plus 4 heme group. 4 protein chains, 2 of them alpha, 2 of them beta, the normal adult, and 4 heme group attached to all 4 protein chains. Those heme group contain iron. That's the structure of the hemoglobin. Now, obviously, when we're talking about oxygen carrying capacity, what happens if we cannot carry enough oxygen? So if we cannot carry enough oxygen, so we cannot meet the demand of oxygen, so oxygen carrying capacity is reduced. We call that a condition anemia. N means like without, so M means blood, yeah means like condition. So it's anemia, like kind of like you have a less contributors so that you cannot carry oxygen. Now it can happen simply, just if we think simple logic, it can happen either you have less red blood cell because you're not producing enough, or you have less hemoglobin, like which you're not producing enough hemoglobin. So less production can cause the problem. 2, loss can cause the problem. Let's say you have enough blood, but you're losing too much. Even the menstrual loss could be. So it's the hemorrhagic. So you're losing that. Or number 3 is destruction. You're not losing, but you are destroying them. Okay, some kind of disease can causing the destruction of the number of blood cells or the protein, whatnot, and that can cause the anemia. So these are the 3 ways it can happen, but anemia can be classified multiple different ways. So what I basically did, I listed the major type of anemias and individual reasoning. Okay, in this course you must know all of these. So aplastic or hypoplastic anemia is when your erythrocytes and hemoglobin synthesis is decreased formation. So we don't make it any. So basically what happens results from defective bone marrow because of radiation, like nuclear radiation, toxin, drug use. All these things can cause, even poison, that can cause the reduction of the production of those blood cells, which lead to anemia. Iron deficient anemia. We know we can use iron to make a heme group. If we do not make a heme group, we cannot make a hemoglobin. So obviously we'll have a deficiency in iron can lead to anemia. Pernicious anemia, deficiency of vitamin B2, B12. The deficiency of vitamin B12, what happens is vitamin B12 is essential for DNA synthesis and all this production of the blood, that molecular level. So vitamin B12 is necessary. But in vitamin B12, when we absorb from food. We don't make vitamins in the body, we take from food. When you eat food, your stomach secretes a factor called intrinsic factor. That factor helps to absorb vitamin B12. So if you don't have that factor, if you're lack with the intrinsic factor for any reason, it could be autoimmune disease or it could be a genetical reason, that if you do not have that intrinsic factor, what are you going to have? You're going to have lack of vitamin B12, which will interfere in the blood production, thereby anemia. That's called pernicious anemia. But it's very similar one is called macrocytic, where the macrocytic one, when the size become big, that's why the name macro, caused by deficiency in folic acid and vitamin B12, both of them, both when they're caused by both deficiency, folic acid and vitamin B12, that's called macrocytic anemia. That's why normally when a woman gets pregnant, they need to maintain the amount of blood, doctor will prescribe the iron tablet, folic acid, vitamins, all these things because of this reason, so that we do not want to cause any less blood production because of this simple reason. Hemorrhaging, losing blood. So either could be diseases like ulcer or any big damage in your body, or as simple as like during reproductive age of women, menstrual cycle. That can also cause the hemorrhagic anemia. And finally, there is a there is a genetic disease, genetic level disease called sickle cell anemia. So where the shape of the red blood cell become like a sickle when the oxygen concentration decrease, and then obviously they cannot carry enough oxygen to the tissue, that caused by a defective production of the beta chain. So remember, hemoglobin has 2 alpha chain and 2 beta chain. So some people who have lack of that allele, like genetic information, they cannot, defective genetic information, they can produce a defective beta chain, which can cause the red blood cell to collapse into a sickle when the oxygen concentration is low, and they can cause a severe anemia to those people. Remember, I mentioned earlier that it's like a leasing a vehicle. So for RBC, it's like a leasing a vehicle, means your body produce them, you using them, and then you recycle them. Body produce them, using them, and then anything wear and tear, they keep an eye on them, and they recycle them. That's how they do it. This is how to do it. So I'm going to use this. I'm going to use this slide to explain how to do it, and then I'm going to go back and explain how they produce. So let's do the recycling first. So how they're recycling. So this is a red blood cell roaming around in your body in the blood plasma. So these cells, those are in the liver or in the spleen, they call macrophages. So they can be in the spleen, in the liver, or they can be in the bone marrow. Macro means big, phage means eater. Remember phagocytosis in NP1? So phagocytosis. So what they do, they actually phagocytose, means they engulf those damaged red blood cells. So after 120 days average lifespan, they engulf them, they eat them, they take them inside. When they take them inside, they break them. So when they break, the main component of the red blood cell is the hemoglobin. So that hemoglobin molecule, if you remember, they have 4 polypeptide chain, 2 alpha, 2 beta. And at the end of each chain, there's a heme group attached. So they separate them first. So the globin part, or the polypeptide chain, they break into amino acid, and obviously amino acid body can easily recycle to make protein again. Whether they make hemoglobin or any other protein, they recycle amino acid. So this can be used for protein formation, even in case of RBC. Now the heme group, which is the non-protein moiety, which is the non-protein molecules. They have mainly 2 parts, an organic part and iron atom. So they literally pluck the iron atom, they separate the iron atom out of the heme, and they recycle them straightforward. So they take away from the heme group, they take away the iron atom, and they carry the iron atom into the bone marrow again. Remember, blood production takes place in the bone marrow, and they're recycling them. And sometimes if you have excess, they store them temporarily as well in the body. So the third variety is the remaining organic molecule, which is the heme molecule, the organic heme molecule. They convert into biliverdin first, which is a little bit greenish color pigment. And then biliverdin molecule is converted into another molecule called bilirubin, yellow color pigment. And then they either carry them to liver, if they're outside the liver, or if it is inside the liver, they are in the liver. Then using the bilirubin molecule, what liver does, liver produce a molecule called bile, B-I-L-E, bile. They produce the molecule called bile. What bile does, bile require for fat digestion. So in the digestive system chapter, we are going to learn that bile emulsify fat. Without emulsification, that's a necessary step. Without emulsification, fat cannot be efficiently digested by the enzymes. So emulsification of fat is taking place in the small intestine where the liver secrete the bile and which made from the bilirubin molecule. And these are the metabolic products of the liver which can either be eliminated in the feces or some of them we can reabsorb in the system and some of them can go into the kidney and eliminated in the urine. That's why urine and feces both appears like a yellowish color. That is why if you have a liver problem, like any disease associated with liver, such as jaundice, can cause yellowishness of your body, like your nail, your body, your eye all turn yellow because this bilirubin is, the red blood cell is breaking down, too much bilirubin in the blood that causes yellowness. That's how they recycle all the material of the blood. So when the blood cell is under wear and tear, they are dying off after like their job is done after 100, 120 days, macrophage take them in, break them, amino acid, heme, amino acid recycle, heme, take out the iron, recycle, heme, change into biliverdin, biliverdin change into bilirubin, and then they transport. So bilirubin is the one that transport to the liver, and the liver use that bilirubin to make bile. Okay, detailed function in the digestive region that I just mentioned, we are going to repeat that in the digestive system again. But when they transport the iron, the protein that does the job called transferrin. So the protein that transport is the one called transferrin. And when you have excess iron, body store them. They just don't throw them out. They store them. They store them by the protein called ferritin and hemosiderin. Okay, so iron is removed from the heme and then transported. The one that transporting is called transferrin. The protein that storing them in the liver or in the spleen, the storing them is called ferritin and hemosiderin. We don't want to throw them because we may need them in future. So that's how the erythrocyte is being recycled in the body. So let's go back one more time. So when we talk about the lifespan and circulation, the average lifespan is approximately 120 days. They replaced before they break down, called hemolysis, and we now know how they did it. They stripped the iron, they converted biliverdin, and then to bilirubin, then take it to the liver, and we did the whole thing. So iron is recycled and excess is stored. Production. So how we produce them? We produce them in bone marrow, mentioned earlier. So how we produce them? The method is called, once again, hemopoiesis. Poiesis means production. And heme, the word came from blood. So blood cell production begins with the hemopoietic tissues. So the stem cell that they produce we call hemocytoblast. And that produce two line of stem cells. One is called myeloid stem cell. The other was called lymphoid stem cell. So myeloid, myle means bone marrow, and lymphoid is the lymphoid organ. So myeloid stem cell, which is happening in the bone marrow, is the parental cell that's going to produce the RBC. And lymphoid stem cell mostly responsible for lymphocyte that we're going to mention later on. Okay, now this is the one we are interested right now. So from myeloid stem cell, so hemopoietic stem cell, then myeloid stem cell. From myeloid stem cell, they produce the erythrocyte. That phenomena, that part, we call erythropoiesis. Erythropoiesis, red blood cell production. Erythropoiesis is regulated, or you can say they speed up the process by a hormone called erythropoietin, EPO in short. That erythropoietin is a hormone that produced by kidney. So in the endocrine system, we learned that kidney is one of the gland as well, and they secrete hormone. So one of the hormones called erythropoietin, in short EPO, which regulate the erythropoiesis, the production of the red blood cell. Now, what is the production? So from myeloid stem cell, so from hemopoietic tissue, or hemocytoplast, we have myeloid stem cell. That myeloid stem cell produce proerythroblast. That proerythroblast then convert into erythroblast, and then basophilic erythroblast, and then polychromatophilic erythroblast, and then polychromatophilic erythroblast become normal blast. At that stage, when they're normal blast, they eject the nucleus. We don't need that nucleus anymore. Whatever protein they made, they already made it, or their process of making from the mRNA that they produced. So they don't need the nucleus. So they throw them out. So we don't need the nucleus, so they throw that nucleus out, they become reticulocyte. So normal blast is the stage when they throw the nucleus out, they become reticulocyte. That reticulocyte come into the circulation. That from the bone marrow, they come into the circulation, and within approximately like 24 hours, they become like a mature red blood cell. So you got to know these series of steps. So from proerythroblast, different stages of erythroblast. The first one is basophilic erythroblast, then polychromatophilic erythroblast, then normal blast. At the normal blast stage, boom, they eject the nucleus. What they become? They become reticulocyte. Once they become reticulocyte, they can come into the circulation. They have produced enough hemoglobin molecule, or they are in the production from the mRNA they already produced, and then they become mature red blood cell within like 24 hours. This is known as RBC production, or phenomena called erythropoiesis, regulated by a hormone called erythropoietin. The erythropoietin is produced by a gland, kidney. We already know the recycling, then we are going to go into the leukocyte. So in RBC, you must know RBC structure. You should know hemoglobin structure. We should talk about anemia and different sets of anemia. Then we know how RBC is produced, then how RBC is recycled, all of this. Now leukocyte, leuko means what? So leukocyte, white blood cell, in 1,000. That's in millions. Now it's in 1,000, like 5 to 10,000 RBC, WBC per microliter of blood, normal. So normal adult here 5,000, but it can go up during the infection. So because remember, main job as a whole, their main job of leukocyte or the WBC is to defend your body. Okay, so then they do have nucleus, unlike RBC, which does not have nucleus. They do have nucleus. So WBC in their lifespan, they stay sometimes in the blood and sometimes they migrate out from the blood. So when they migrate out, so from the blood vessel, so let's say this is a blood vessel, and that's what the problem is. So right here on the wall, that's your skin, and this is your blood vessel. So the problem is there. So if the body comes to know that there is a problem, those WBC from the blood vessel, they go out. So they literally squeeze out from those capillary. So between two capillary cell, if they put a little bit of, if they're between two capillary cells, if they get a little bit of space, they can squeeze out. That's what they call amoeboid movement. They're capable of amoeboid movement. Remember amoeba? They can change their structure to squeeze out. That's what they do. So they go towards the margin of the blood vessel. So this is the vessel. They go towards the margin. So at the end of the cell, and then they squeeze out. So when they squeeze out during amoeboid, and they follow positive chemotaxis. What does that mean? The chemotaxis means wherever the injury site, let's say that's a skin, that's what the injury is, those cells release molecules. Those molecules attract its WBC. That is what the movement called chemotaxis. So I'll give an example. For example, if you keep a plant near the window, the plant slowly, when they grow, they bend towards the window. Why? They're looking for light. So wherever there's sunlight, they're bending towards the sunlight. That's called phototaxis. Taxis means movement. So photo, towards the light they move. In this case, they're moving towards the chemical, wherever the chemical is releasing. So those released chemical attract them, chemotaxis. And then when they go there, some of them, they literally are capable of phagocytosis. Phago means eating, so they're eating the microorganism or any damaged cell. That's how they save you. Now they have many different types. They have 5 different types of WBC. We can actually make them into 2 groups. One group we call granulocyte. the other group called agranulocyte. The follow this name. The granulocyte means their cytoplasm has many granules. So, excuse me. Their cytoplasm has a lot of granules. So if I show you a picture right here, you see this right here, those are the granules we're talking about. In this case, their cytoplasm has a lot of granules, different color, but still granules, a lot of granules. But this appearing slightly cleaner than this, and this is almost very clean. So these two are agranulocyte. These three are called granulocyte, meaning the cytoplasm has granules in it. Those are lysosome, like lysosomal granules. Okay? That's what the two name. So granulocytes, they are neutrophil, eosinophil, basophil, three types. Agranulocyte, two types, lymphocyte and monocyte. Now, one more time, let's go back to this picture. So in neutrophil, eosinophil, and basophil, these three are granulocyte. The monocyte and lymphocyte, these two are agranulocyte because they don't have granule. Now, even if you focus the word like neutrophil, eosinophil, and basophil, so what happens is their cytoplasm of these cells, their cytoplasm has the philicity, the liking towards something. So this you can figure out easily. Basophil, meaning their cytoplasm is basic. So they take the basic dye, basophil. Their cytoplasm is acidic. The dye itself called eosine, that's why it's called eosinophil. Some book may refer to them as acidophil. This is basophil, this is acidophil. Meaning their cytoplasm is acidic, that's why it's appear red or pink in color. So under microscope, if you look at a WBC slide, you'll find some eosinophils are like reddish or pinkish in color right here. This one, this one, like pinkish in color. This is another picture just to show you. Probably this is more real life than this is a little bit exaggerated color here, but still they appear like this. Neutrophil, their cytoplasm is neutral, neither basic nor acidic, neutral. They're going to appear like obviously neither pink nor like blue. They're kind of in between. That's one difference. The second is this nucleus is a little bit different shape. So when you look at all the 5 cells under the microscope, if you see a fragmented nucleus, by the way, this is one nucleus. They're connected individually. That's why they're like a fragmented. It's not nuclei. It's one nucleus, but they're like fragmented. They have 3 to 5 lobes. Those are called neutrophils. Clearly, they're connected, but these are 3 to 5 lobes. They're called neutrophils. It is a bilobe, like 2 lobes, narrowly connected in the middle, like 2 lobes, narrowly connected in the middle with a pink dot, like a pink dye. Those are eosinophil and acidophil. That's how we can recognize. Basophil, most cases you cannot see the nucleus very well. The reason is they're kind of behind too many granules. So when you cannot see the nucleus properly, like too many granules, those are basophil. Monocyte is the largest one, the big, and they have like a kidney-shaped nucleus, or like upside down U or kidney-shaped nucleus. They're big. Lymphocyte, on the other hand, they're very small, the smallest one, and it looks like a tennis ball. And 90% of their area is like covered by the nucleus. So they have like a tiny ring of plasma membrane right there. Those are lymphocytes. So these are the 5 variety of WBC. So when a clinician taking your blood and counting like your blood, they are actually figuring out how many neutrophils you have, how many eosinophils you have, how many basophil, how many monocyte and lymphocyte. That gives us a direct information that what may going inside your body. So in a normal blood, neutrophils is like 60%. Then second one is lymphocyte, is approximately 30%. Third one is monocyte, is approximately 8%. Then the eosinophil and then basophil is very, very less. But that's our normal distribution. So it was an old mnemonic like N, so that's how it's never let monkey eat bananas. So never let monkey, eat bananas, neutrophil, lymphocyte, monocyte, eosinophil, basophil. That's their distribution. Now, if that change, that is not fixed, that can change based on your infection. So let's say you get infected with some virus or bacteria or some other pathogen or whatnot, that will change. So based on that, like, like 2-page detailed discussion of your book. So I would give you this suggestion: read one time of your book and then go back to this particular slide and read them like 5-6 times to understand each one what they do. So the neutrophils, they're responsible mainly for like bacterial infection. So, and eosinophil, that's like a lot of allergens, like allergy time. For example, I have like pollen allergy in the springtime, and in the fall time, I have some other type of allergy. That's why my throat get always choked. So I probably have tons of eosinophil in my blood all the time, most likely, particularly in the spring, 100%. Then basophil, they are increased in number of the chicken pox, sinusitis, diabetes, and lymphocyte. They increased they're like main immune response cells. So they are also monocyte. They are also phagocytic cells. So again, you read them and you try to memorize individually what they do. And this is how they look like. I give 2 slides intentionally. There's 2 different pictures just to get an idea of what they look like. So Leukopoiesis, production of the white blood cells. Remember, hemopoiesis is the production of blood cells. Is 2 different ones: erythropoiesis, leukopoiesis. So erythropoiesis does it from myeloid stem cell. Leukopoiesis, they also do it from pluripotent stem cells. So some of them are called myoblasts, some of them are called monoblasts, and some of them are called lymphoblasts. They are technically known as like CFU, colony forming unit. The colony forming unit, meaning they do a separate set of cells. Okay, in your book, you may have a nice chart to explain which one goes where, but here you have to memorize that myeloblasts, they form neutrophil, eosinophil, basophil. Monoblasts, so myelo means they're mainly in the bone marrow. Monoblasts, they form monocyte. Lymphoblasts, they produce lymphocytes. Okay. Without going very detailed, this is what you have to memorize. Platelets. Platelets technically is not a cell because it's like a fragment. So megakaryocyte, mega means big, karyo, the word came from nucleus, and cyte means cells. So cells with a giant nucleus, let's say this big of a cell, they get fragmented. So they just blow up themselves and produce a lot of fragmentation. Those are platelets. They're like, appears like a flattened disc, and they're very small and fraction, and they're different sizes in their blood sphere. They circulate approximately like 9 to 12 days before their phagocytes remove them, but they have a main function. One of the major function is clotting. So blood, remember, we have a fixed amount of blood. We have an amount of blood, and we are replacing them, but we have that amount, like 4 to 6 liters. So let's say average 5 liters of blood, and they're roaming through all your blood vessels. Now, if you get a damaged blood vessel, blood will come out. You don't want to lose too much. If you lose too much, anemia is the first thing, and then obviously other things will kick down. Okay, because your brain is continuously depend on the nutrients. So if you don't have enough blood, you cannot supply nutrients to your brain. So you do not want that damage to continue. So these platelets come into the function first. They first try to clot, like create a platelet clot, which I'm going to describe a little bit later, and try to minimize that loss. So approximately you have 130 to 400,000. I mentioned earlier, but this is micro. Something happened to the symbol. That's microliter of blood, not liter of blood. So they have microliter, like a drop, like 130 to 400,000 platelets per microliter of blood. Now the numbers, and we talked about it. So there are certain words referring to the numbers. So the basic word is this: penia and cytosis. If you look at the word called penia, penia means deficiency or poor, cytosis more. So leukopenia, less number of WBC in your circulation for whatever reason. Leukocytosis, high number of WBC. So let's say you have infection, obviously you'll have leukocytosis, that's temporary. Thrombocytopenia, deficiency in platelets, thrombocytosis, high in platelets. Same thing, erythrocytopenia, which is we also call anemia. That's another word, like deficiency in RBC. And erythrocytosis, which is also known as polycythemia. Polycythemia is basically, poly means many, cyte means cell, hemia is blood cells. So we have many blood cells. We have polycythemia is basically excess RBC. And other than that, leukemia is the blood cancer. So leukemia is when you have a very high number of WBC. So let's say like 80,000. So when your WBC number skyrocket, like 80,000, 90,000, that's blood cancer. That's when whole white blood cell, they're proliferating without any control. That blood cancer. Leukemia is the blood cancer. So leukopenia is a low blood WBC. Leukocytosis, a high WBC, could be regulated. But leukemia is the blood cancer. That's like when not only high, that's skyrocketed. So instead of 10, they're like more than 80,000. So that's what all the numbers are. So you should note that. Then final two things. One is like hemostasis, the blood clotting. The other one will be the blood typing. So blood clotting, very important factor. I already explained why. Because we do not want to lose the blood. So it has three phases. So it's like your blood is like garden hose. So they're flowing through a pipeline. What is the first phase? It's called vascular phase. So what happens, let's say you are using garden hose to watering your plant. And if it leaks, what we do instantaneously before we run and close the faucet, we squeeze the hose just to minimize the loss. That's exactly what body is doing. That's called vascular phase, meaning when the blood vessel is carrying the getting the blood, as soon as they get a damage, the blood vessel can damage, they crack open not only from outside, from the inside, we have a lot of injury all the time, and that can happen. And as soon as the blood is coming out, that vessel will constrict, squeeze. There are chemical reaction that allows the vasoconstriction. That's called vascular phase. So that will minimize, but that will minimize for a limited amount of time, okay, because it's going to go back normal again. Then come platelets. So that's a second phase called platelet phase or platelet clock phase. What that is, like platelets, they normally do not, when the blood vessels is smooth inside, if the inner wall is smooth and not damaged, they have a layer of like prostacyclin. So that prevents the platelets to engage or get stuck with the side wall. They don't, they just keep flowing. But as soon as there is a damage, platelets generate like a pseudopod. Pseudopod means like extra pod means feet. The pseudopod, they will stick to the damaged area and then they keep sticking like this. So when there is a damaged area, they keep sticking like this. So they created a plug so that they minimize, they bring those two damaged portions, like they pull them together so they can minimize the damage. They're not very strong. Obviously, too much flow will make it open then. But that's what the first, second stage of hemostasis, trying to lose the blood. That's why when you have a cut, you don't want to wipe the blood. You want to blot it. You want to keep it like this because more you wipe, you're removing those platelet plugs, so you are causing more bleeding. So if you want to stop the bleeding, you should just press it like this. Keep it pressed until the third phase kicks in. That is called coagulation phase. That's where the coagulation takes place. Now, what is coagulation or clotting? In this case, mainly referred to as clotting. What is this? Just imagine this. We have the platelet clots. We have a lot of RBC, a lot of WBC there. What we need something like a cheesecloth. Just imagine, we need something like a cheesecloth. If they can hold those platelet and the RBC and WBC together like this, by the time body will slowly repair the damage, then it's fine. Like that clotting process is called coagulation phase. So basically, we have everything, we just generate a cheesecloth. So it is better understood if you look from the bottom up. method. So what basically it is, I'll explain from this slide. So let's go back one more time just to give you an idea. This is the cheesecloth I'm talking about. That's a net. Cheesecloth means net, which is grabbing all the cells. Could be red blood cell, could be white blood cell, could be platelet plug, and they are just keeping it right here, so that the vessel that got damaged, sorry, can get time to repair, so that blood cannot get lost right here. Okay, so after platelet plug, they create a coagulation net right there. So that is the network. Now, how they did? So this is where I wrote down, but I'm going to explain this from this picture. Okay. So there is a protein called, by the way, this coagulation process requires, like, a lot of them are discovered yet. Your books have mentioned some of them. It requires like more than 30 factors. It is one of the most complex phenomena that we studied in our body chemistry. One of the most complex, because you have to understand, as soon as it gets damaged, the blood should clot. But they cannot continue clotting, because then there is a bigger problem. So they should clot and then stop clotting immediately. So it's a very complex thing goes on. There are so many factors. We actually call them factors. So most of them produced by the liver. Okay? So factor, and we name them like factor 1, factor 2, factor 3, factor 4, factor 5, and so on. Again, for this class, you do not have to memorize all the factors. You have to just know a few that I'm going to mention. So please pay attention. So this one, the protein called fibrinogen, which is the clotting protein we studied, that is in the blood plasma. They are soluble. The fibrinogen is a soluble protein, meaning they are roaming around with the blood plasma, because they're soluble. Once they convert into a protein called fibrin, that's their insoluble version. That's an insoluble strand. As soon as the fibrin produce one strand, the other strand, and they can kind of like together make a network of strands. There are other factors involved, but they can make a network of strands. That's the cheesecloth I'm talking about. That's the net I'm talking about. That net, once they produce, they can capture all the cells to create that clot. So bottom line is, if fibrinogen is there, no problem. As soon as it converts to fibrin, it can clot. And it does clot. Now, who converts them? The enzyme is called thrombin. But our blood should not have thrombin in an active form, because if they do have an active form, they will clot the blood all the time. You don't want blood to be clot all the time. So thrombin is produced only that time when the clotting is required, because of prothrombin, that inactive enzyme become thrombin. Now, who makes them inactive? See that? Like a series. So who make them inactive to active? There is an enzyme called prothrombinase. So the question is prothrombinase in the blood? No, it can be, because if it is, then it will clot. So then prothrombinase is there, but it's in the inactive form. We call them factor 10. Now, who activates factor 10? Because somebody has to activate factor 10. Once the factor 10 is activated, they can do this, got it. But somebody has to activate factor 10. There are two pathways, there are two series of reaction, two cascade of reaction that can activate factor 10. One is called extrinsic pathway, one is called intrinsic pathway. Now, let's understand the word first. Extrinsic means the factors that are responsible initially does not come from blood. It's outside the blood. It could be from the blood vessel, which is considered outside the blood, the cells in the blood vessel. Intrinsic means those factors are in the blood. Intrinsic. Extrinsic means outside, intrinsic means inside. So it actually begins with extrinsic first, and then immediately after, it's the intrinsic take care of it. So there are many factors. The first factor, that the one I wanted to know, is called tissue factor. As soon as they get a damage in the tissue, means the wall get damaged, tissue get damaged, they release the factor. factor 3, commonly called tissue factors. And this tissue factor can activate other factors to initiate the intrinsic, and they can continue to the other factor to initiate the extrinsic as well. You do not need to know this series of reactions. You do not need to know this series of reactions. You do not need to memorize all the factors. What I want you to know, the tissue factor is the first factor that comes out of the tissue damage. You need to know there are two pathways, extrinsic and intrinsic, namely. Both of them are capable of activate factor 10, which initiate the common pathway, because factor 10 then activate prothrombinase. Basically, they activate it, become prothrombinase, then prothrombinase activate prothrombin to thrombin, thrombin change fibrinogen to fibrin. You know this. And you should also know that calcium ion, which is one of the factors, and vitamin K is also one of the factors, which is not mentioned in this slide. So calcium and vitamin K are the two very important factors for the blood clotting. Okay, so that's basically this. Now we have a regulation, because as I say, we cannot just keep clotting. The regulation is a lot of detail. Even in your book, they mention some of it. So what I did, I make it very simple. I make you remember just three components. Okay, antithrombin 3, heparin, and thrombomodulator. These are the three molecules that are directly involved. There are others. Don't go into that very detail. So antithrombin 3. So we know thrombin, the enzyme that convert fibrinogen to fibrin. So antithrombin. Follow the name, antithrombin 3. So antithrombin, they inhibit thrombin directly. So they're controlling the clotting. Heparin, they accelerate the antithrombin. So heparin is telling antithrombin, hey, can you stop the thrombin, so that they cannot make the fibrin. That's exactly what they're doing. Then there is a molecule called thrombomodulins. They are modulators. They inhibit several clotting factors, like different clotting factors that are involved, plus they stimulate an enzyme called plasmin. Okay. So what does plasmin? They convert from plasminogen to plasmin. What is that plasmin thing? It's called kallikrein, another name, which basically digests the fibrin strand. Remember, once the fibrin strands happen, they are like a clot. You don't want a big, huge chunk of clot inside your blood vessel. Why? Because later on, think about the blood flow can rip that up, and that clot can continue. And if it is a big clot, because it happens in a bigger blood vessel, it can stuck to a smaller vessels. It could be in your heart, could be in your brain, and clot that vessel cause, like in the brain, it can cause a stroke. Then the heart, it's a myocardial infarction or heart attack, it can cause, because they will block the blood vessels, the narrower one. So we need to digest that. That digestion of fibrin is called fibrinolysis. Lysis means breaking down. So you're breaking down fibrin. And who does it? Many things. One of the main enzyme is plasmin. So this plasmin is stimulated by thrombomodulins again. So that's a little bit of regulation of blood clotting. So now we are at the final stage, which is the blood types. Blood types, I'm sure you heard about that, like type A blood, type B blood, type ABO, and all these words. But what are these? Like, this is the part you got to understand. Okay, just have to understand. So there are two words we commonly use: antigen and antibody. Antigen are the molecules. Okay, they reside on the cell surface. Okay. Now, there are foreign antigen also. Antigen is a big word. Basically, nowadays we call antigen that can refer to or that can generate an immune response. So a lot of foreign molecules, like even bacteria, virus, different other pathogens, can act as an antigen as well. When they go over, even like the pollen can act as an antigen, because when they go in the body, they can generate an immune response. It can happen. Just make it simple right now, without going into that detail. That blood cell, they carry some molecules attached to their cell membrane. Those molecules directly have no function to the blood cells function, meaning they are not responsible, or they are not changing the oxygen carrying capacity of the blood. I'll give you an example to understand this. It is like you're painting the outside of your house. Now, let's say you and your friend or brother or mom or sister got a very identical house next to each other. And they're all like townhouses lined up, like 10 of them. And you bought one, you decided to paint outside the pink. Okay, that's a pink house. But that doesn't change the house because all the houses, same model, same design. The other one, somebody decided to paint white or blue. That's it. That's just identification, recognition. So those antigen molecule is just for recognition purpose. The other cell can recognize them. So if my cell has one particular variety, so I know, okay, pink house is my house. That kind of thing. So antigen, there are some antigen molecules. These are particularly glycolipids. They're attached to the red blood cell. Okay. Now the enzyme. Now the enzyme that are producing them are also controlled by the genes. So for those antigen need to be is the product of certain genes. So those are in your DNA. So based on your genetic information in your blood, you have certain type of antigen. That's what I want to say. Okay. On the other hand, antibodies are some protein molecules, that gamma globulin that we studied, that body produce. They have a series of process. The plasma cells in the body, they produce this. We are going to learn in lymphatic or immune system class. Right now, you just need to know antibodies are the gamma globulins of the proteins. They react against antigen. That's good enough. How they form, you don't need to know. So they react against antigen. And finally, the word agglutination or neutralization. The agglutination when antibody attack the antigens. So they agglutinate. In other words, like they cause clumping of those cells. So because they attack from multiple directions. So it looks like when sometimes you have a milk that goes bad, like stale milk. The casein comes out. It looks like that, like when the blood is coagulated. Okay, so this is the three word you need to understand. Antigen is a molecule that are attached to the blood cells. They do not affect the function of the blood cell. They're just recognition purpose. Antibodies produced and they then attack the antigen. Agglutination is that attack. So this is an example. So in the body we have two main antigen called A and B. Okay, this is how you can understand, because O meaning these are the precursor molecule. From here we make either A or we make either B. This is the precursor. That's a main molecule. This is not really an antigen, but this is a molecule we need. Okay, without going very detailed, we need to make either A or B. So if you have an enzyme to make A, you're going to make this. If you have an enzyme to make B, you're going to make this. When you have this, that means you have nothing. Consider you have nothing because that's a precursor. That's not a proper antigen. So, but they don't change the function of the blood cells. Now the things is very simple. So if your blood cell has only the precursor, meaning nothing, I'm considering precursor means nothing, then we call it type O blood. If you have a type of antigen, we call type A blood. If you have a B type of antigen, we call type B blood. And if you have both the type, like A and B, we call AB blood. So I'll give you an example, simple example. Let's say if you have a phone which has like Verizon, so you're a Verizon subscriber. If you have a phone with AT&T, you have AT&T, you are AT&T subscriber. Let's say we have these two subscriber only. And if you have enough money to have one Verizon and one subscriber, okay, you're AB type blood. You have both the variety. That's it. Doesn't change anything. Phone, no matter what phone you have, when you speak, we speak the same thing. We talk. It doesn't really matter. And if you have, let's say somebody has no money, so no phone, that's O type. That's it. So you do not have a cell phone, you're O type blood. You have in Verizon, you're A type. You're AT&T, you're B type. You're both Verizon and AT&T, you're AB type. That's it. That's as simple as this. Now, when you have two type of antibodies found in the blood plasma, okay, body produce them. So if you have there are two types, anti-A and anti-B. These are the two types of antibodies. So now anti-A, follow the name, anti-A means they work against A. Anti-B, they work against B. That's why the name. So anti-A will attack A, anti-B will attack B. Now if you have an A-type blood, you will have anti-B antibody in you. Anti-B, because you don't want to attack yourself. You want to attack if somehow your blood gets mixed with the B type, then you're able to attack them. So if you have an A-type blood, you're going to have anti-B antibody. If you have a B-type blood, you're going to have anti-A because you don't like it. So it's basically, you are a Verizon customer, so Verizon will sign up with you. They say, oh, you cannot move to AT&T. If you want to move, then you have to give $500. Same thing with AT&T will do. That's exactly the same thing. Now, if you have a type AB, if you have both the phone, you really don't care about what antibody you have because nobody's protecting you. So you have neither A or nor B antibodies in your body. And if you have no cell phone, that means you have both the antibody, meaning you cannot get any one of them. You have resistance. So you cannot get anti-A, you cannot get A, you cannot get B. You have both the antibodies in your body. Because when you have the opposite antibody, you're going to attack, you're going to agglutinate. This is what exactly happened. So that's your basic blood type. Type A blood means you have type A antigen and anti-B antibodies. Type B blood, you have B antigen and anti-A antibodies. Type AB blood, you have both A antigen and B antigen, no antibody. Type O blood, you have no antigen, but you have anti-A and anti-B, both antibodies. That is why when you have an A-type blood, you cannot take from B-type, because if you take blood from B-type, your antibody is going to attack them. You cannot take blood from AB type because your antibody is going to attack them. Do you remember? That's what they are also B. But you can easily take blood from O type. So if you have an A type blood, you can either take blood from another A or O. If you have a B type blood, you can take a blood from B or O because nobody is attacking O. And if you have an AB type blood, nobody can take from them. They can just take from themselves. And they can take from B, and they can take from A, and they can take from O. They can take from nobody can take them. They can take everyone because they don't have anything to attack. So they have neither anti-A nor anti-B antibodies. So they cannot attack them. That's why they are called universal acceptor. They accept everybody. On the other hand, O can only take from O because they have both the antibodies. So they don't like any other blood. So they can give to others. So they're universal donors. So O can give blood to everybody, but can receive only from O. AB can receive only from AB. Can give nobody, but can receive from anybody. Type B can only receive from B and O. Type A can only receive from A and O. So you have to write it down, and you have to understand this logic behind to understand what blood typing is. And finally, there is another antigen got discovered. It's called Rh factor. They discovered in a group of monkey called rhesus monkey. That's where the word is Rh came from. So there are multiple varieties. Out of them, the C variety, D variety, E variety, but D is the one that's more reactive. So that's why we call them either D antigen, or simply call Rh factor. So that's a third variety. Apart from A and B, now third variety, T-mobile. Either you have it or you don't have it. If you have it, we call positive. If you don't have it, we call negative. So let's say you have a Verizon phone, as I give you examples. So go back to here. You have type A already. And on top of that, you have T-Mobile. So we call them A positive. If you don't have the T-Mobile, we call them A negative. Same thing with AT&T subscriber. If you have it, we call B positive. If you don't have it, B negative. AB positive, AB negative. O positive, O negative. So finally, O, they can at least use the T-Mobile, O positive people. So ABO is a group, and then comes RH on top of it. RH is also known as D antigen. So some book will refer to as D antigen. So you can have two choices, either you have or you don't have. If you have, you're positive blood towards them. If you don't have, then you're negative. Most people have it. Like 85%, if I remember correctly, Caucasian people or 99% Asian people are positive. There are few in every group, they're negative blood, okay, meaning they don't have RH factor. Now what happens is when you don't have RH factor, those women, when they get pregnant, I'll come back to this 2 seconds later. When they get pregnant, those women, the first pregnancy, because the mother is RH negative, so during the first pregnancy, let's say the baby is positive. If they're negative, that's not a problem. If they're positive, it could be A positive, could be B positive, could be AB positive, or could be O positive, any of them. Because normally, if the mother is A, pay attention to this one. If the mother is A, forget about positive, negative. If the mother is A, the question you have to ask yourself, can the baby be B? Because mother A blood means they have anti-B antibody. Can the baby be B? The answer is yes. Why? The answer to that question is anti-A and anti-B antibodies, they cannot cross placenta. So if they cannot cross placenta, they cannot attack the baby. The baby is fine. But the problem comes when the mother is negative. Whatever, A, B, or O, doesn't matter, negative. What happened then? First pregnancy, not a problem, but during either the first pregnancy get like some kind of abortion happens, or let's say during the regular delivery, when the placenta breaks, the mother's blood get mixed with RH factor. Now those RH factor, when they get mixed with mother's now immune system is going to make anti-RH antibody. So when these anti-D or anti-RH antibody, anti-D is another name of RH, is in the mother's body now. Now when they're pregnant for the second time, so not the first pregnancy, the second pregnancy, when they're pregnant for the second time, now RH antibody, they can travel placenta, unfortunately. And if the baby is positive blood, if the baby is negative, it's not a big deal. If the baby is positive blood, those RH is going to attack the baby's blood, and they cause severe anemia and a toxic brain syndrome. This phenomena we call hemolytic disease of newborn, HDN, hemolytic disease of newborn, HDN. So that's what happened when a RH negative mother have a RH positive fetus twice or more. Nowadays, obviously, so this is what exactly I said, they can cross placenta. Nowadays, normally it does not happen anything because there is an injection called ROGAM. They give the pregnant RH negative woman. So basically what happened is immune system, when they generate antibody, they memorize it. We're going to learn in immune system chapter. In this case, before mother's body produce the antibody, because it takes some time, and they destroy it. So therefore, the mothers never, like as if the mothers never got infected, or I'm going to use the word infected, they never got mixed with the anti-RH antibodies. Okay. Now going back one more time, right, this is the normal drop of blood. The agglutination reaction that I was talking about, this is how we do the blood testing. So this is the normal blood sample. Now in the blood sample, let's say like this is, let's say, Mr. not Mr. X, Y, Z, and P, like 4 people, different people. So you put all 3 drops here, and then you add anti-A serum, anti-B serum, and anti-Rh serum. And you look for which one they reacted. And if they reacted to the anti-A and anti-Rh, that means this blood sample has A antigen and Rh antigen. So we call them A plus blood. Remember, having A antigen means A blood, having Rh means plus, so A plus blood. Similarly, this antigen react with only B and Rh, we call B positive blood. Then they reacted with all 3 of them, AB positive. This reacted none of them. None of them reacted, we call them negative. They neither have A, nor they have B, nor they have Rh factors, call them negative. That's how we do the blood testing. So that is basically the entire chapter. So the blood chapter, you have to first go through the individual components. So as a whole, the blood and individual components as I described. So individually, you need to know what they do. So, and then first plasma, then you, obviously in the plasma, you look mostly, you talk about protein, and then you go to RBC, WBC, platelets. So in the RBC, you learn not only the structure of the RBC and hemoglobin, you also learn production of the RBC, recycling of the RBC, and detail about anemia. And then in WBC, you have 5 different types, 2 groups, granulocyte and agranulocyte, 5 different types. Not only you need to know the name, but also you need to know what individual one of them does. And please pay attention to the slide on that. And then platelets, same thing, they're responsible for blood clotting. And then finally, all the numbers like polycythemia, anemia, leukopenia, leukocytosis, leukemia, thrombocytosis, thrombocytopenia, all these things, numbers means go up and down, like what happened. And then comes your hemostasis, meaning blood clotting. You need to know the vascular phase, platelets phase, and coagulation phase, and finally blood typing. Okay, so read the book first, and then watch the lecture video with the PowerPoint slide, and then re-watch them, and that's it.